Hello everyone. So today we are going to look into different dimension reduction techniques in the context of single cell RNA sequencing data. So we have seen previously that single cell RNA sequencing data is very much high dimensional, and dimension reduction methods are actually very important pre-processing methods that are often applied on the single cell RNA sequencing data. So the single cell RNA sequencing data are again very much high dimensional, so we have seen this before. So measurements corresponding to thousands of cells can be obtained in a single experiment. So in fact, depending on the technology, the number of cells can also vary. Nowadays, there are very high throughput technologies that can even profile up to 100,000 or even million cells. In fact, a very recent study came out just few days back, which actually profiled around 4 million cells from the tissues. And it's also very much high dimensional with respect to the number of genes that are being measured from the single cell. So in fact, each single cell is a very high dimensional data vector. So there can be measurements corresponding to 25,000 genes. Again, it's an approximate number, so the number of genes can also vary depending on the organism and depending on the technology. But in short, we can see that all these data sets are very, very high dimensional. So the question is, how can we actually utilize this high dimensional data in order to answer specific biological questions? So it turns out that even though these data sets are very high dimensional, there actually exists an intrinsic low dimensional structure for the data. So even though around 20,000, 25,000 genes are sampled or they are profiled, only a subset of those genes will be actually present in the asset sample and measured with non-trivial variability. So of course, we are measuring genes on the order of 20,000, 25,000, but only a subset of those will be actually meaningful in the data. And on the other hand, cells actually do not have completely random gene expression values. In fact, these gene expression values are not evenly distributed across all the dimensions of the data. So maybe some dimensions are actually more important as compared to others. So in fact, we can see that many genes, they have very highly correlated expression value across cells. So this is one type of structure that we also talked about when we discussed the imputation method. So essentially, there is information redundancy in the dataset where multiple genes have collinear expressions. So essentially, the effective dimension of the data is much smaller than the total number of genes profiled. And in fact, we can say that the single cells are actually occupying a lower dimensional manifold. So this lower dimensional structure of the data is also known as a lower dimensional manifold, and this is also sometimes known as the phenotypic space or the latent space of the cells. So in fact, the distances that you can measure in the high dimensional data and the clusters that you can obtain from the high dimensional data, they are also preserved in the low dimensional structure. So this is particularly an experiment where people actually measured the correlation between the high and low dimensional distance. So the y-axis over here actually represents the correlation value, and these are measured for different types of experiments, some single cell and some bulk RNA sequencing data. And they have observed that as the number of measurements increases, the correlation actually becomes much better. So even with 400 genes or so, the correlation almost nears 1. So you can see that all the distances, again, the distances are basically the properties of the data. The distances in the high dimensional data and the clusters, they are also preserved in the low dimensional structure. So that's why we can actually use some sort of dimension reduction technique to map the single cell high dimensional data to the lower dimensional structure. So let us look into some of the dimensionality reduction techniques which are actually existing in literature. And in fact, dimension reduction techniques are very common problems in machine learning, in the domain of machine learning, where oftentimes you encounter different high dimensional data, such as in video or images and so on. And of course, single cell RNA sequencing data is an example of a high dimensional data. where multiple different dimensionality reduction techniques have been applied successfully. So informally, dimension reduction technique actually deals with a transformation that results into a reduced dimensional structure as obtained from a high dimensional data. So basically it actually transforms, and this sort of techniques actually transforms the high dimensional data into a lower dimensional data, but the lower dimensional data must have a meaningful representation. So this reduced dimension representation that is obtained by the dimensionality reduction techniques should have a dimensionality which is corresponding to the intrinsic dimensionality of the data. So what do I mean by this intrinsic dimensionality? Again, you can think of intrinsic dimensionality as the minimum number of parameters that are required to account for all the different observed properties of the data, of the high dimensional data itself. So of course, the number of dimensions in the original data set is much high, but essentially this reduced representation, if it actually corresponds to the intrinsic dimensionality of the data, then it can actually represent all the observed properties of the data efficiently. And in fact, this high dimensional data, dealing with this high dimensional data becomes very, very difficult in different scenarios because of the curse of the dimensionality. And this dimension reduction techniques, after representing the data in the reduced space, it can help us a lot in various tasks, such as it can actually facilitate the classification of the data. So oftentimes dealing with high dimensional data becomes very computationally expensive, whereas the reduced dimension, it can actually facilitate the classification. And of course, another important aspect is visualization. We cannot visualize very high dimensional data, but imagine that we can actually, if we can actually project the data into 2D, then it will be very suitable for visualization, and we can even identify different structures in the data when visualized in 2D. And at the same time, it's also doing some sort of compression of the data into lower dimensional space. So traditionally, dimensionality reduction has been obtained through different linear techniques such as principal component analysis, factor analysis, or classical scaling. Now, such type of linear techniques cannot actually adequately handle very nonlinear high dimensional data which are observed in the real world. So instead, different nonlinear techniques have also been developed which can deal with complex nonlinear data. And in fact, they are much more suitable for real world data such as single cell RNA sequencing data, because the real world data is likely to form a nonlinear manifold. Again, the manifold is again the lower dimensional structure, and the shape of the manifold can be very nonlinear in case of real world data. So that's why this kind of nonlinear techniques can actually handle the real world data better as compared to different linear techniques. So let us formally define this dimension reduction problem. So let's assume that we are given a data set which is represented by this letter X. It is consisting of n data vectors represented by xi, and each data point or each data vector has dimensionality which is denoted as capital D. So it's a very high value, so that dimensionality is high. Now we can imagine that the intrinsic dimensionality of the data is represented as small d. So of course small d is smaller than capital D, and oftentimes it is actually much smaller than capital D. So the meaning of intrinsic dimensionality being small d is that the points in the original data set, they can be thought of as lying on or near a manifold with dimensionality small d. So that is the represented by this intrinsic dimensionality small d. And this manifold which has a dimensionality small d, it is actually embedded in the high dimensional space. So that is the definition of the intrinsic dimensionality formally. Now this manifold, regarding this manifold, we are not going to assume any specific structure of the manifold. In fact, it can be even non-Riemannian manifold because of the presence of discontinuities. So essentially, the manifold can also consist of a number of disconnected submanifolds. So we are not going to assume any particular structure of for this particular manifold on which actually represents the intrinsic dimensionality of the data. So essentially, dimension reduction technique is actually a transformation. So this kind of techniques can transform the data in the high dimensional space into a new data set, which is represented as capital Y. And this new data set has dimensionality d, small d. And while doing this transformation, the dimensionality reduction techniques must retain the geometry of the data as best as possible. So again, by retaining the geometry, I am again hinting towards the structure of the manifold. So essentially, the structure of the manifold or the geometry of the manifold must be retained as much as possible while performing this transformation from the high dimensional data X to the lower dimensional data Y. So there is a problem with this particular transformation or this particular task. So we do not know the geometry of the data manifold. So that is actually unobserved and unknown. And at the same time, we do not have any idea about the intrinsic dimensionality of the dataset. So both these two things are unknown to us. So that's why the dimension reduction problem becomes an ill-posed problem, and we cannot generally solve it. We have to solve it by assuming certain properties for the data, such as the property that we are assuming that it has an intrinsic dimensionality represented as small d. So only under such assumptions we can solve this dimension reduction problem. So this here is a taxonomy of available dimension reduction techniques, and again this is taken from a paper that was published in 2008 or so. So you can see it actually lists the methods that were available at that time, and mostly actually it actually only lists some of the linear techniques, but it mostly talks about the non-linear techniques. Even after this, a lot of new dimension reduction methods have been developed. In fact, after the development of a lot of models, deep learning models, a lot of deep learning models can also perform dimension reduction as is observed nowadays. But in this particular lecture, we are going to focus on some of these methods that have been represented over here. Again, broadly, these dimension reduction methods can be classified into two groups, convex and non-convex. The convex methods actually optimize an objective function which does not contain any local optima. So in such case, it is possible to obtain a global optima, whereas the non-convex one deals with objective functions that can contain local optima. So the convergence to the global optima is not guaranteed for the non-convex dimension reduction methods. So first of all, we are going to look into some of these convex dimension reduction method, and we are going to start with principal component analysis, which is also the most popular linear technique for dimension reduction. So this principal component analysis technique actually performs dimension reduction by embedding the data into a linear subspace of lower dimensionality. So it actually constructs the lower dimensional structure as a linear subspace. So essentially, this lower dimensional structure also describes the maximum amount of variance that is present in the data. So this lower dimensional representation, it is constructed in such a way so that the maximum amount of variance is explained by the lower dimensional structure. So it turns out that PCA is actually attempting to find a linear mapping, which is denoted as M, that also maximizes the cost function given by this, where covariance of X is the sample covariance matrix, and essentially the goal is to find this linear mapping which minimizes this, which maximizes this cost function. So this linear mapping M is actually formed by the d principal eigenvectors of the sample covariance matrix. So this particular covariance matrix, if we can compute the principal eigenvectors. So again, we have to beforehand we have to mention the dimensionality of the reduced space, which is again denoted as small d. So if we can compute the principal eigenvectors of this covariance matrix, then we'll obtain the linear mapping. So essentially these principal eigenvectors, these are actually also called as principal components. So essentially the goal of this principal component analysis technique is to solve the eigen problem, which is denoted as this. So again, we are trying to find the principal eigenvectors of the covariance matrix, and so this is the equation which is actually solved to obtain the principal eigenvectors. So the low dimensional data representations are then computed by performing this transformation. So y is the product of the original data matrix multiplied with the linear mapping. So again, this transforms the data into the lower dimensional space. So principal component analysis is identical to classical scaling, which actually tries to obtain a linear mapping, which actually minimizes the Euclidean distance between two sets of two matrices, where again this Dij is actually representing the Euclidean distance between two data points in the high dimensional representation, whereas this Yi and Yj, this is basically the Euclidean distance. So this over here is the Euclidean distance between the data points in the lower dimension. And of course, this Yi, Yj, these are restricted by the linear mapping, so that's why it is written as this. And there is another particular constraint, which is basically each component of the linear mapping, and the norm of each is actually 1. So this is the setting for the classical scaling, which is trying to retain the Euclidean distance between the high dimensional data point as best as possible in the low dimensional data point. So it turns out that this particular cost function can be minimized by the eigen decomposition of the Gram matrix. Now again, the Gram matrix is nothing but the product of the original high dimensional data matrix and its transpose. So basically this is the Gram matrix, and by minimizing, in order to minimize the cost function, we need to compute the eigen decomposition of this particular matrix. So this is how principal component analysis is very much related to the classical scaling problem. And you can see that essentially the goal is to retain the Euclidean distance between the high dimensional data points as best as possible. Now there are some limitations with PCA. So the covariance matrix that is computed, or the eigen decomposition is computed on the covariance matrix. The size of the covariance matrix is actually proportional to the dimensionality of the data points. So in case where the data is really very, very high dimensional, then it becomes computationally expensive. And the second limitation of PCA is that it is actually focusing on retaining these large pairwise distances, which is given by, again, the Euclidean distances. Whereas if the manifold is nearly really nonlinear, so in such cases it's much more important to preserve small pairwise distances. So again, distances between the nearest neighbors in the manifold. So this kind of retention of Euclidean distance in the lower dimensional data sometimes actually limit the applicability of PCA. So next we are going to talk about a method which is known as Isomap. So again, as I said before, if the high dimensional data actually lies on a curved manifold, which is really a nonlinear manifold, then Euclidean distance will not work very well, because essentially PCA-like technique or classical scaling will actually look into retaining the Euclidean distance. But if the manifold is nonlinear, then the Euclidean distance between the 2 points can be actually misleading, because the actual distance has to respect the geometry of the manifold, and As a result, we can end up with retaining some distances which are not really meaningful as per the structure of the manifold. So that's why this Isomap technique actually tries to accommodate this problem with a new type of distance. So it actually preserves geodesic or curvilinear distances instead of Euclidean distances. So again, the geodesic distance is nothing but a distance which is measured over the manifold. So essentially it is trying to respect the geometry of the data. So let's look into the algorithm itself. So first of all, this Isomap technique will actually construct a neighborhood graph, which is denoted as capital G. So in the neighborhood graph, basically every data point in the high dimension data, every data point is connected to its nearest neighbors, k nearest neighbors. So that is how the nearest neighborhood graph is constructed. And in that neighborhood graph, the shortest path between the 2 points actually gives us a measure of the estimate of the geodesic distance, because essentially the nearest neighbor graph is here only each data point is connected to its nearest neighbors, and there can be many paths through the graph. Of course the shortest path can be computed using Dijkstra's algorithm or any other algorithm, and that will give an estimate of the geodesic distance. And after that, if we compute the pairwise geodesic distances for every pair of data points, then we obtain a pairwise geodesic distance matrix, and on that we can apply classical scaling to obtain the final lower dimensional representation. So this is how this Isomap technique actually tries to accommodate for a curved manifold, again a nonlinear manifold, by computing this or by preserving these geodesic distances in the lower dimensional structure. However, this Isomap technique also suffers from some limitations. So in fact, because of topological instability, Isomap can actually construct erroneous connections in the neighborhood graph. So oftentimes, as I said earlier, that the manifold structure is not really known. So in some cases, there can be erroneous connections because of the unknown manifold, and in such cases, this can lead to some sort of short circuiting in the graph. So essentially, it may look like that the 2 points are very close to each other, but then again, that is really misleading because of the erroneous connections. It can also suffer from some holes in the manifold. Again, this is a specific terminology coming from the topological analysis. And finally, if the manifold is non-convex, then it can actually fail. So it will not be able to recover or obtain the global optima if the manifold is non-convex. So next we are going to talk about kernel PCA. So again, this is actually reformulation of the original principal component analysis method, but in a different space, which is obtained by a kernel function, by applying a kernel function on the data points in the original high dimensional space. So again, the kernel matrix, it's basically is going to compute a kernel matrix, and the kernel matrix will just be very similar to the original data matrix, as it will be another in product. So essentially, this sort of kernel PCA technique is going to accommodate for different types of nonlinear mappings, so depending on the choice of the kernel function. So this was actually proposed in order to get rid of the linear assumptions that was associated with the PCA. So using this kind of using a nonlinear kernel function, it is possible to accommodate for the nonlinearity in the data. So first of all, this kernel PCA techniques actually computes the kernel matrix using the data points. Now the entries in the kernel matrix are defined by this equation. So again, it's a k kappa over here is a kernel function, and it has to be it can be any function which actually leads to a positive semi-definite kernel. So a lot of different types of kernel functions can be applied, such as Gaussian or some sort of polynomial kernel can be used. In fact, if the kernel is linear, then it basically giving rise to the PCA. So this kernel PCA can be thought of as a more generalization of the PCA technique. After that, the kernel matrix is double centered. So essentially, it is double centered using this particular equation. And in fact, this double centering is actually equivalent to subtracting the mean of the features in the traditional PCA. So finally, the kernel matrix, because of this double centering, it actually becomes the zero mean data. And after that, on the centered kernel matrix, first of all, we are going to compute the principal D eigenvectors, which are again denoted as Vi. So now the original, you can remember in case of PCA, the linear mapping was obtained by solving an eigen problem where we obtain the principal eigenvectors of the covariance matrix. Now in this case, we are actually first computing the eigenvectors of the centered kernel matrix. Now how do we obtain the eigenvectors of the covariance matrix then? So it turns out that they are related using this particular equation, where this is eigenvector of the covariance matrix and this is the eigenvector of the centered kernel matrix, and this is the eigenvalue of the corresponding eigenvector. So to obtain the low dimensional data representation, this particular data is projected onto the eigenvectors of the covariance matrix, again which is obtained by this particular equation over here. So as you can see, this again the kernel function is being used to project the data onto the lower dimensional space. So now, which kernel function is actually going to be useful? There it becomes ambiguous. It can vary from one problem to another. So this particular technique, maximum variance unfolding, it actually attempts to learn the kernel matrix on its own. And in order to do that, it first is going to define a neighborhood graph of the data. And again, so just definition or building this neighborhood graph is very similar to Isomap. That's what we have seen. And it at the same time, it actually tries to retain the pairwise distances in the resulting graph. So again, the first task it performs is to construct a neighborhood graph G, and in which the data point is connected to its k nearest neighbors. So this step is very much similar to what we have observed in case of Isomap. And then it actually attempts to maximize the sum of the squared Euclidean distances between all data points. But at the same time, it is actually looking for a constraint so that the distances inside the neighborhood graph are preserved. So while it is actually trying to maximize the Euclidean distances between all the data points, at the same time it is also preserving the distances in the neighborhood graph. So somehow it is trying to basically retain the structure of the neighborhood graph, and that's how it is learning the manifold somewhat. And essentially this is the particular optimization problem that it solves, right? So essentially this it is trying to maximize the Euclidean distances between the data points in the lower dimensional space, subject to the situation where basically their distances must be the same coming from the neighborhood, in the neighborhood graph, whatever is existing. And in order to solve this, they actually use semi-definite programming. So it turns out that this kind of MVU techniques are also, they also suffer similarly as in case of Isomap. So next we are going to talk about a technique which is known as diffusion maps. Now you actually came across these diffusion maps when we talked about the method magic for imputation of the data. So these diffusion maps are based on defining a Markov random walk on the graph of the data. So again, we are constructing a nearest neighbor graph of the data, and on that graph we are defining a random walk, which is again a Markovian random walk. So if we perform this random walk for a number of time steps, then we obtain a measure of proximity for the data points. And this actually defines the diffusion distance. And the concept behind the diffusion distance is basically that it actually integrates over all the paths through the graph. So because it's actually performing the diffusion process in the graph itself, and the diffusion process is nothing but a Markovian random walk. So after these diffusion distances are actually obtained, then we can perform different tasks. So essentially the goal of the diffusion map technique is to retain the pairwise diffusion distances in the lower dimensional representation of the data. And in order to do so, first it actually constructs the graph, the nearest neighbor graph. And in the graph, the edges are also weighted. So these weights are actually computed using the Gaussian kernel function to construct a new matrix W, where each of the entries are actually denoted as or given by the Gaussian kernel function as applied on the original data matrix, high dimensional data. And again, sigma is actually a parameter of this Gaussian kernel itself, which controls how basically the value of sigma will be controlling how the Gaussian kernel, how wide the Gaussian kernel is. After that, we perform row stochastic normalization on the matrix W. So essentially, it's normalized in a sense that each row, the sum of the entries in each row becomes 1. And this kind of normalization leads to a new matrix, which is denoted as P superscript 1. So entries of this particular matrix are given by this. Again, you see that this is performing just the row stochastic normalization. And this is actually our transition matrix, the Markov transition matrix. So this matrix gives us the transition probability from going from one data point to another data point in a single time step, because this is actually the transition probability for a single time step. Now we can actually think of this particular transition probability at probability matrix as the random walk forward probability matrix for a single time step. If we want to compute the probability matrix for t time steps, which is again denoted by P superscript t, it is actually given by the matrix exponentiation of the single time step transition probability matrix. So again, the exponentiation is over with respect to t, which is t is the discrete value which gives us the number of time steps. Then the diffusion distance between two data points is actually given by this particular equation over here. So you can see it is actually summing over all possible paths, right, in the based on this random walk, which is consisting of t time steps. So again, in this particular equation, in the denominator, this actually is computed like this, where mi is actually corresponding to the degree of the node, but it's actually summing up all the probabilities associated with the edges, right? And essentially this attributes more weight to the denser part of the graph. So it actually adaptively computes the diffusion distance. So essentially the parts of the data points which has very high transition probability, forward transition probability, they will have a small diffusion distance. So now we have to actually recover our lower dimensional representation, which is again denoted by y. And as I mentioned earlier, diffusion map, the goal is to retain a lower dimensional representation which preserves or retains the diffusion distance, again which is given by this between two data points, diffusion distances as best as possible. Now it turns out that this lower dimensional representation is obtained by the non-trivial principal eigenvectors of the eigenproblem, which is again given by this. So we have this forward transition probability matrix for t time steps, and we want to find the non-trivial principal eigenvectors of this by solving this eigenproblem. So it turns out that basically the graph is actually fully connected. So that's why the lower dimensional representation is given by this. So we are starting from the second eigenvectors and all the way up to d plus 1. Again, d is our dimensionality for the lower dimensional data. So the graph is fully connected, so that's why the largest eigenvalue is trivial, which is basically 1. So that's why we discard it. So again, it has been shown that the lower dimensional representation is given by this the d non-trivial principal eigenvectors. So this is how diffusion maps actually solves the problem of dimension reduction. And again, by performing this or by computing this diffusion distance, which is again summing over all possible paths or integrating all the possible paths between two data points in the neighborhood graph, it is actually more robust as compared to the geodesic distance, which is computed by the Isomap technique. So these were all the techniques that actually construct a low dimensional representation by performing an eigen decomposition of the full matrix. So these are all known as full spectral techniques. Now there are other kind of techniques which are also known as sparse spectral techniques. So instead of performing an eigen decomposition of the full matrix, again full matrix referring to the complete data matrix in the high dimensional space, these sparse spectral techniques are going to solve a more sparse eigen problem, which is again a generalized version. So essentially these sparse spectral techniques are going to focus on the local structure of the data, and it will try to retain the local structure of the data in the lower dimensional space. So in the next lecture, we are going to take a look into these sparse spectral techniques. Thank you.